Hello, everybody. I hope you're all doing well. I was really impressed with your MQ7 work, and I realize that MQ8 is, you're thinking, well, it's all done now. I want to kind of um, put your mind into a date in the future, May 18th. May 18th, 2 in the afternoon. This is a Monday after you um, graduate. But you still have this. You still have this two free response questions in order to get credit for the class. And I'd ask that you give it a good go. So I've been hitting MQ7. I did a lot of free response, and I want to keep doing them. I'm going to try to make them fairly low work and high grade. Low work, high grade. What a nice concept. But I do ask you to use your neurons there. Think your way through, because at some point, May 18th, 2 o'clock, you'll be asked to be using your own brain material. These two questions are the latest round of free response, and I chose them for a distinct reason. This first one is, what if you had a question that you don't know nothing about, you can't remember it, oh my gosh. Well, that's why we do these. We kind of just refresh our memory. And this thing just says, explain the role of each numbered step. So let's use a few catchwords. This is obviously a phospholipid bilayer. This is obviously a membrane-bound, even a transmembranous protein. This is obviously the hormone that they're referring to. It says a generalized hormone. But remember that video that we watched with the Bozeman science guy, the camp? There's several series of steps there, and this is kind of similar to that mechanism, but we're not asked to explain all those different specifics. Really, it's just each step. At step 1, you have a hormone bound to a membrane-bound protein. And sometimes they call this a second messenger approach, or a two-messenger approach to hormones. This is your first messenger, this membrane-bound hormone. And the second messenger is where this protein here activates a protein in the cytoplasm to create some sort of response. And that you would be writing that, second messenger, or protein signaling a second protein floating in the cytoplasm to create some sort of change. At step 3 here, we have this same hormone going through the nuclear envelope, through even the pores of the nuclear envelope. Show the reader that, hey, you know what you're talking about, even if you don't. You probably know a lot of these things. The nuclear envelope has got pores in it. But then we have a protein here, and it's obviously next to DNA. And what is it doing next to that DNA? Well, it's affecting the expression of that DNA. What kind of watchword could we use there? How about transcription? It's affecting the transcription of a gene that would respond to this initial hormone over here. Just try to use a lot of catchphrases and catchwords and biological terms that you've used, and fill out a paragraph. This kind of problem here is, again, a long time ago, and just think about what you know is really what I'm trying to say there. Think about what you know and expand on it. This is the other one. The free responses, I imagine, will be fairly difficult. And one of the free responses, the first one, is 25 minutes long, and it's supposed to be interpreting and evaluating experimental results, which is why I put this one in here. So you've got you've got a map, you've got the ocean, you've got Connecticut, you've got Long Island, New York City's in there somewhere. Um, and if you just look at this, this table, you can obviously see that as the sites go from 1 to 8, the salinity goes up. The salinity goes up from 1 to 8. And that makes sense because this is the ocean and this is land, And if you read this, I mean, the reading gets horrible. So, they're talking about leucine aminopeptidases and things like this. But the real question says, based on the data, describe the most likely effect of salinity on the frequency of the lap allele in the marine mussel population. It's simple. The sites go from 1 to 8. The salinity goes from low to high, and the lap allele frequency also goes from low to high. And that would be your answer, and I would kind of expand on it. Why do you say that? Because the allele frequency is 13 at a low salinity site 1, and it goes up to 16 at site 2, and so on and so forth. Show that you actually have eyes and read the table. Simple, really, though, isn't it? I mean, this is horrible. This is written by some Albert Einstein freak, but really the question is quite simple. And then it says, predict the likely lap allele frequency between site 1 and 2. Okay, what's the answer? 13 to 16. Site 1 is 13, site 2 is 16. So the frequency of alleles, I imagine those are percentages, 13% or 16%, somewhere between 13% or 16% based on the table above. People, I'm going to keep hitting these free responses, and I'd ask that you do them, show some effort to them, and practice makes perfect. I think if you keep practicing on these, you got a good shot at getting a good grade on the AP exam. So I'll catch you later. Bye bye.